I'm guessing they figured that you guys will ask all the good questions, so they just sit at home and watch it on the tube. Okay, so let's get started. Just one announcement, and of course this will go out to everybody in TV land. I have written the exam. The exam has 150 points, and the breakdown of points is that Section 1 has 32 points, and the Section 2 has 90 points, and Section 3 has 28 points. So that's the split of the points. Is that right? Let's see. 28, 120. Yeah, I think that's right. I think it's right. Yeah, 32. That should add to 150. Yeah. Why do I think in my head that doesn't add to 150? Is that 140 or 150? That's 30. Oh, no, 32. That's right. Okay. Yeah. Yeah. Doing math in my head. All right. So I hope your studying is going well. It's kind of weird we won't have class for the first time in 2 terms on Monday, so that's a little odd. Relief, right? Yep. So where should we start? Or should we start and just going home? Yes, sir. I didn't hear the. So I heard a coding region, and then what? So when I say the term coding region, does it refer to RNA or DNA or both? Well, technically it's both. The code is actually read from the RNA, the messenger RNA. But when I talk about a coding region, the DNA, since it codes for the RNA, is a coding region as well. So that term is used interchangeably. Liz?
Can you explain splicing and the whole?
Yeah, can I explain splicing? You like that figure the book gives you? I don't like that figure at all. Yeah, I'll talk through splicing a little bit. Let's do that. So, and I've had some confusion I've heard raised from students about the nature of what's involved in that splicing. So, and part of it I think is my fault. So let me hopefully clarify that for you a little bit. And I didn't realize this until I had a couple students ask me about this. So let me talk about what's not splicing, and then I'll talk about, to answer your question, more about how splicing occurs. Okay? So when I talked about prokaryotic systems, I talked about tRNA and ribosomal RNA processing. You might remember this figure. And I've had a couple students a little puzzled by this. They wanted to know where the exons are. Okay? Well, this isn't a spliced message, okay? So and maybe this is going to be a little confusing, but it's not spliced. This is a processed message, all right? So what this is the way that transfer RNAs and ribosomal RNAs are organized in prokaryotes. So prokaryotes make one long RNA that contains all these various genes. I shouldn't say genes, all these various regions for ribosomal RNA and transfer RNA. Then, this guy is literally cut into pieces. So the difference being that if I cut, I might make 50 pieces out of this RNA. All right? The difference between that and splicing is all I'm doing here is cutting it into pieces. I'm not rearranging and joining the pieces back together. Makes sense? With splicing, I'm cutting, and I'm cutting sections out and then rejoining the sections around the pieces that got cut out. So this is simply a processing. All right? So the thing that comes to my mind is if I had a long piece of wood and I chopped it into 50 pieces, I could use those 50 pieces to do something with. All right? But if I wanted to make something big, I would take those 50 pieces and say, here's a rotten section. I'm going to leave this out, but I'm going to join this good section to this good section. Now I've got, a longer piece than either of the 2 pieces started with. So this is not that. This is simply processing. And this processing that you see happening here happens also in eukaryotes. Eukaryotes do something similar to this as well. And that's not splicing. So it's not splicing. Splicing will always involve the removal of internal pieces. Right? So let's take a look at splicing. Okay. So splicing will always involve a piece that's removed. The piece that's removed is called an intron. And the pieces that get joined together are exons. So in this splicing process, this inner portion is removed. This AG of the blue gets joined to this G of this green, and we have a spliced messenger RNA. Okay, the term dates back to the old days when we used audio tapes, and that if you wanted to edit an audio tape, you had to physically take a pair of scissors and cut out the section that you didn't want, and then you use a piece of tape to join together the pieces that you did want. Okay, and it was called splicing. That's where the term actually comes from. And you can do the same with videotape. That's how they used to edit movies, doing the same thing. All right. Now, your question had to do with how does this occur? So how does this process occur? It occurs in a mechanism that is, as I said, not the best figure here. All right. In general, your book has pretty good figures, but in splicing, it doesn't have very good figures. What's happening here? Here's the initial RNA that's been made. Now, most people talk about this as a messenger RNA. This actually could be a transfer or a ribosomal RNA. Okay? So I could have taken one of those pieces that I chopped out of a eukaryotic big RNA, and then this might be a ribosomal RNA, and it has introns within that one of those 50 pieces. Okay? Doesn't matter. All we have is an RNA that's got things in it that we want to remove. The things that we want to remove are the introns, and the introns are excised in this fashion. Okay? Here's the longer RNA that's there. This particular guy has 2 exons. It could have 50. There are actually eukaryotic messenger RNAs that have over 100 exons. Okay? Complicated? Yes. And some of the problems that arise with some of the diseases that we see probably relate to some of the splicing problems. Okay? There's a protein called dystrophonin, which is implicated in human muscular dystrophy, that has about 100 exons. It's enormous. It's spread over about 1 million base pairs, and a lot of crap has to get cut out of it. And so there may be some problems related to the size of that gene. All right, getting back to this. Here's exon 1, all right, and here is exon 2. Here's the piece that's going to get removed. Now, as I noted in class, there are 2 things that are absolutely found in every intron, and that is that the very first base of the intron is G followed by a U, and the very last 2 bases of the intron are A followed by a G. Okay? Almost always, there is a pyrimidine-rich region. That's what this Y stands for here, pyrimidine, any nucleotide, pyrimidine, purine, A, pyrimidine. There's usually some region about 30 base pairs away from the place where the splicing is going to occur that has this. The A that has this side hydroxyl group is participating in the splicing mechanism. The A has a 2' hydroxyl because it isn't attached to anything. That 2' hydroxyl will make a nucleophilic attack on the phosphate between the G of the exon and the G of the intron. That causes a break to occur and is what forms ultimately the lariat structure. So here's a covalent bond that has been formed as a result of this nucleophilic attack. Remember, this is occurring in spliceosomes. Spliceosomes are those RNA protein complexes that do the splicing. So we're inside of a spliceosome here. The spliceosome has made this attack. The 2' hydroxyl has made the attack inside of the spliceosome. And we have made this lariat structure. Well, the second part of the process involves the attachment of this G, which now has been rotated up, that's why I don't like this figure, has been rotated up here, okay, to make a nucleophilic attack on this phosphate. All right. That nucleophilic attack causes this guy to become joined, which is what happens here. We see now this blue has joined to the green, and that leaves the RNA, the intron, left out, and it's left out as a lariat. Right? That's a lot of words, a lot of words. As I said in class, I'm not overly concerned with the details of mechanism here. Okay? This is not like we're doing serine proteases and we're very concerned how catalysis occurs. What happens in this process is that the intron gets removed as a lariat, the two exons get joined together. Question over here?
So is like the SNRNAs,
Yes.
when they form these pairs with like, the protein-rich area and the
Yep.
is that what basically kind of catalyzes this kind of?
Yes, it's a very good question, and you're exactly right. This figure, in fact, there's not really any good figures that your book has, but probably the best figure that your book has on splicing is actually shown right here. Okay? This shows a little bit more clearly, at least, how the snRNAs are participating in aligning this stuff. And what I like about this figure is you can see that with the base pairing that's set up here, okay, these are snRNAs. Here's the intron. There's that pyrimidine-rich region that's here. And you'll notice that the complement over here doesn't include a pair for that A. And so that physically causes the A to stick out. And it's the strain and the sticking out on the A that makes it an attractive OH to make a nucleophilic attack right there. So that's the geometry. Just they don't have a good geometry figure because in reality, this guy right here has to be brought into close proximity to this thing right here. Well, why couldn't they just put the two like that? And that's really all they have to do. And the figure makes sense. But it is, you have to do sort of mental gymnastics with your head thinking, okay, now this guy's free and now it has to flip over here. And that's not really what happens. It doesn't flip like that. There's another hand. Did you have a question?
What happens to the intron? Is it just kind of
Yeah. So people always ask about that. It's interesting. So the intron is mostly junk. Most of the introns, probably 99% of the introns, get chewed up by a nuclease, and then we get the salvage pathway for the nucleotides to reuse those nucleotides. However, there's a very, very tiny percentage of introns that actually have protein coding, believe it or not, in there. And so they can actually be translated in a few cases, okay? But for the most part, they're just simply degraded. Yeah.
Sorry, how does the sRNA do that? Like, does it just create that A and it attacks it? But like, I could have sworn sRNA had like another.
Well, there are several snRNAs. Okay.
Okay.
So that's why I say this is a simplification of the process. There's I think there's 4 or 5 snRNAs that are involved in the overall spliceosome, and they each have their individual functions. There are also snRNAs, if I recall, that'll also do some pairing out over here. Okay. But I'm not sure I understood what your question was.
Yeah, I don't really know.
Okay. Yeah. So, but the snRNAs, as I said, there's many proteins here. This is all set up, and really, it's all set up to make this geometrically, very easy to do. Why your book doesn't show you that, I don't know. So. Yes.
the difference between the cancer sequence and I think operon in coli.
Okay.
That because in the cancer sequence there may be distinguished, right? Many different sequences. But for operon, some of them are caused by a singular sequence.
Okay. So let me just, I'm not sure I understand your question, but let me just address that. So he's asking about enhancer sequence and operons. So there's no relationship whatsoever between enhancer sequences and operons. So enhancer sequences are things that we find in eukaryotic DNA, okay, that facilitate the transcription of genes in tissue-specific manner in eukaryotes. Operons are sections of prokaryotic DNAs in which genes are clustered together on a single messenger RNA. So I'm not sure what you're asking me, but there's no relationship between those two terms whatsoever. Okay? Now, one difference that I did note, maybe this is what you're thinking of, is that in eukaryotes we don't have operons. Every eukaryotic gene is on a single messenger RNA. We don't see multiple genes on one messenger RNA. In prokaryotes, we see that. And that's one very fundamental difference between the two. Okay? So enhancer sequences allow eukaryotes to do tissue-specific transcription. And, of course, for a prokaryote, they don't need tissue-specific because they don't have any tissue. They've got all the cells are the same.
So can you talk about siRNAs?
Well, can I talk about siRNAs? I can, but I haven't talked about them during the term. So there's not a good reason to do that other than to confuse people.
Am
So.
I just confusing
You're confused. You're thinking about microRNAs?
Yes, that's
Okay.
what I'm confusing them with.
Okay. So microRNAs, though they do have a somewhat similar function to siRNAs, are not siRNAs. So siRNAs are not things that I've talked about this term. But to answer your question, let's take a look at the microRNAs that I did talk about. And there's really only one figure that I had here, and that's unfortunately the only figure that I have relating to microRNAs. But microRNAs are short RNAs that are present in eukaryotic cells. They're typically transcribed by RNA polymerase II. I'm not sure if I said that in class or not, but if not, you can use it. I'm not going to ask you that if I haven't asked you about it. I haven't said it in class. But they are typically about 60 nucleotides long or so. Okay? And they have a section of them that's complementary to a target gene in that eukaryotic cell. So let's say I have an insulin messenger RNA, and the cell might have a microRNA that would be complementary to a portion of that insulin RNA. And because it's complementary, they can form a base pair. The base pairing that occurs between that microRNA and the target gene, RNA, in this case the insulin, comes together in a protein structure called argonaute. And what argonaute does is it says, oh, here's a double-stranded RNA, I'm going to cleave it. And so it cuts the RNAs, and the result of that cleavage is that it basically either makes the messenger RNA non-functional, or, depending upon where the cut occurs, it may shorten its lifetime. So in either case, both of these things will affect how much of that given gene is being made. Now, it's a very wasteful process when we think about this. The cell has gone to the trouble of making a complete messenger RNA. And only after it's made that complete messenger RNA does it make something complementary to it and says, oops, I don't want to make that much of it. Okay? But that's what it does. Yeah. And this mechanism is not an unusual one. There are, I think I mentioned in class, something like 700 human genes that are known to be regulated by microRNAs. So it's not a trivial number. Does that answer your question?
Yeah. And I have to ask this question, even though I know I asked it in class, but what's its difference with siRNA?
So what's the difference with siRNA? Okay. All right. Well, the difference with siRNA is that siRNAs are made as a short sequence, and they are made with an inverted repeat. Okay? So, in other words, let's say that an siRNA has a, so here's a, let's see, here's a microRNA. My artistry is not going to show very well here. Here's a microRNA. Here is an siRNA, and then it has a region like that. This is about 60, okay? This overall thing works out to about 22 in the end. What happens is this is complementary to this. So this folds back like so, okay, and makes something that's about 22 base pairs in length. This is, 44 in length, and it folds over like this. Whereas this is this size. This doesn't have a foldback inside of itself. Okay, so rather than waste time talking about something that you're not responsible for, I'll talk to you about that separately.
Okay.
Okay. Yes, Giovanna.
Other than the Scheindel-Garneau, can you go over again the differences in translation between prokaryotes and eukaryotes?
Certainly. So differences in translation between prokaryotes and eukaryotes. And she said, except for Scheindel-Garneau, and I'll just mention Scheindel-Garneau briefly as one of those, because Scheindel-Garneau is a very big difference between the two. So when we look at a prokaryote, all right, one of the reasons that we have differences in translation, or at least the differences that we see in translation between prokaryotes and eukaryotes, relates to the fact that prokaryotes have operons. Now, I haven't really discussed this, specifically in class, but since you've asked the question here, I'll bring this up. Operons have multiple genes on a messenger RNA. Okay, so we saw that when this ribosome comes along to the messenger RNA, it says, okay, I'm going to assemble, and I'm going to assemble right around this start codon because I know where to assemble because here's the Scheindel-Garneau sequence. it assembles, and we go through that whole process. It moves, it translates, it hits a stop codon, and what does it do? It comes apart. But now there's another gene over here. Okay? So in the case of the prokaryote, they have to have multiple places where they can assemble ribosomes. So they have multiple AUGs, but they also have to have multiple Shine-Dogarno sequences to tell the ribosome this is a gene, this is a gene, this is a gene, this is a gene, etc. Okay? So that Shine-Dogarno sequence is very important in prokaryotes, because in prokaryotes, they've got multiple genes on a messenger RNA, and that alignment has to happen each time that a ribosome assembles. In a eukaryotic cell, that assembly only has to happen once. Okay? And as I said in class, one of the big differences between prokaryotes and eukaryotes is in eukaryotes, usually the first AUG is the one where translation starts. So that assembly can happen anywhere along here, and then when that AUG pops in, okay, or whether let's say it slides along until it gets to that AUG, then that assembly can start. It doesn't have to worry about which one to hit, right? Okay, so that's a big difference between the two. Another difference between the two relates to the cap and the messenger and the poly-A sequence that's on the eukaryotic messenger RNA. Prokaryotes don't have caps. They don't have poly-A sequences. Okay, and I showed a figure in class that pointed out that those two. This is gene expression. It's wrong. Those two structures can form a circular structure. Okay, so in eukaryotes, we can form this circularization. Okay, all right. So that happens as a result of interaction between proteins that recognize the cap and proteins that recognize the poly-A. Well, it turns out that the cap and the poly-A both have functions in translation, and primarily those functions are this circularization that you see here. And it appears that the reason that cells do this is to keep the ribosome components, which are disassembling over here, in close proximity to the AUG that's over here. Okay? So by keeping the end close to the start, the cell is able, this is not to scale obviously, the cell is able to more efficiently translate those messenger RNAs. Okay? Now, the only other thing that I said was different about eukaryotes compared to prokaryotes relates to the very first amino acid. Okay? In eukaryotic cells, the very first amino acid is methionine. In prokaryotic cells, the very first amino acid is called 4-methionine. It's got that 4-mel group on it. And there are other slight differences, but nothing significant enough for our purposes, and nothing else that I've talked about. Okay? The names are different. You can see the names different up here. But yeah?
How much information do you want us to know about the T1 receptors, the sweet ones, for the mice?
Okay. Well, I'd say anything that I talked about in class. So I talked about that experiment that showed fairly convincingly that you had to have those, I think it's 2 and 3 together. The mice had to have 2 and 3 together to make this heterodimer in order for their ability to test sweetness. So yeah, whatever I talked about in class there. Jared?
Would it be possible for you to review the tryptophan operon?
Tryptophan operon, sure. Yeah, tryptophan is always the one that gets the most questions. So the attenuation of the tryptophan operon, which is what Jared's asking about, is always harder to explain than the actual phenomenon itself is. So let's go back to the structure here. Okay. Now, actually, I'll tell you what. Let me see if I can find it. I've got another lecture, a different textbook I use. Let me see if I can find that for you guys and show you this other figure. You can see what I'm talking about with this. Just a second. Okay. Enzyme controls. Synthesis. Remember where that is. Must be. There it is, right there. Okay. That's not what I want. Okay, so this now shows you the tryptophan operon, I think, in a better way. If you find this confusing, don't worry about it because you're not responsible for it. But I like this better. Okay, so what we can see is that there are really 4 regions of this operon. 1, 2, 3, 4. Okay, so we can have regions 1 and 2 paired, and we can have regions 3 and 4 paired. Or we can have regions 2 and 3 paired, and if 2 and 3 pair, 1 can't pair with 2, and 3 can't pair with 4. That's what I was referring to in class. Okay? Now, what does this mean? If this ribosome is moving along and it is getting through this region fairly fast, it gets over here, 1 gets to pair with 2, 3 gets to pair with 4, there's a terminator, it kicks out. So if the ribosome makes it through here rapidly, then termination happens. It'll get through rapidly when there's plenty of tryptophan. If the ribosome gets over here and is looking for tryptophan, oh, wait, no tryptophan, no tryptophan, I'm waiting, it gives 2 a chance to pair with 3. And when 2 pairs with 3, 3 can't pair with 4. So there's no termination. So that's a much better figure, I think. Your book sort of omits the fact that these alternate pairings can happen, but it's a very important part in understanding the operon. Does that make more sense?
Yes,
Yeah. So basically, the whole thing comes down to the abundance of tryptophan. All right. The cell, and people always say, well, what happens when it runs out of tryptophan? Well, if the cell ran out of tryptophan, it's dead. Okay. So the cell is never out of tryptophan. It may have low concentrations of tryptophan, but by the time it runs out of tryptophan, it's dead. It's like if it runs out of ATP, it's dead. Okay. It's not like it's going to all of a sudden start up ATP when it has none. Okay. So all these are how much of anything that it has. So we're measuring the amount of tryptophan by the rate of movement of the ribosome. That's basically what we're doing. The faster the ribosome moves, the more tryptophan there is. And the slower the ribosome moves, the less tryptophan there is. Well, if there's less tryptophan, we want to make the proteins to make the tryptophan. If there's plenty of tryptophan, we don't want to make those proteins. Other questions?
I have a quick question about how RNA is synthesized.
Okay.
You're talking about how there's like a bubble that's created.
Yep.
I mean, but when DNA was synthesized, you had a leading and a lagging strand.
Yep.
Does RNA always get synthesized in like the leading strand kind of way? Like it always
Okay.
gets added on?
Well, if you think about it, in RNA, there's no reason to make a lagging strand.
Yeah.
So to answer your question, you have a bubble. You actually have a bubble in DNA as well. We draw it as a replication fork, and that's what we call it, but it actually is a bubble of sorts. But in DNA, you need to duplicate both strands. There's no reason to copy the other strand in RNA because that's just going to be gibberish.
So will it always open in the way that it creates a leading strand then?
So,
Because I mean, if you have like a piece of DNA, you need to open it like in either one direction or in the other direction.
that's correct.
So will it always open in the way that RNA will always be synthesized from 5' to 3'?
Okay. So what you're asking is, how does it know which way to go?
Yeah.
Okay. So let me remind you of this. Okay. If we look at a promoter, okay, and here's the consensus sequence for promoter. Let's just focus on this for the moment. What's the sequence of the other strand?
The same thing but opposite.
Okay. So we have A, T, T, A, T, A. This points this way. This points this way. This is not the same sequence as this is.
Okay.
So it knows which strand is which.
Uh-huh.
If this were the same sequence, that is, if I had GAATTC, when I read it the other direction is GAATTC also, it wouldn't know which way to go. But because this is asymmetric, it knows which way to go. So this is actually an orientation thing. It's important that it's not symmetric. Asymmetry actually tells it which strand to do. Liz?
Is the region at negative 35 with TTGACA also an AT-rich region?
Yeah, the region at minus 35, an AT-rich region, it is also, yes. It's not quite as conserved as the minus 10 region is, so that you don't see quite the same homologous sequences or comparison of sequences between different genes. So yes, it is fairly AT-rich, but it's not. That's not as important. In fact, it's not really as important because at minus 35, you're not really unwinding much back at that point. This is AT-rich here for the unwinding purpose.
So like at the negative 35 here, if this region resembles this one perfectly, you're going to get more transcription.
The more at the minus 10 matches this, the more transcription you're going to get in a prokaryotic cell.
Is that the case at negative 35 as well?
No,
Okay.
no.
Is the negative 35 she's speaking of in prokaryotes?
Yes.
Does it have any special function that the negative 10 one doesn't?
Okay, so if we see conserved sequence, what would that tell you in the first place?
That it's very important.
That it has an important function, okay. And so that it's not there randomly, okay. So what did this sequence do? Why was this sequence? What was the function of this sequence right here?
That sequence was so that it could open up easily so that you could start transcription.
Okay, so it's partly there so it could open up easily. That's the AT-rich part of it. But there's another reason that this sequence is here. What is it?
Eggs start here.
What's that?
Like the eggs start here.
Yeah, exactly. So it's a place for the sigma factor to bind. So the minus 35 is also an important part of contact for the proteins that bind to start transcription. So specific sequences will play roles because they're recognized by specific proteins to do specific things. Okay. So just like we saw in protein structure when we see conservation of amino acid sequence, when we see conservation of structure, you saw some good examples of that with the histones. I told you that when we looked at those 4 histones, they all had very similar structure. It told us that structure was very, very critical because if we change that structure, that protein isn't going to function. Similarly, when we start seeing conservation of sequence, that tells us that that sequence is going to be doing something very important. And almost always, that importance lies in the fact that there's a protein that binds to it. Giovanna?
So I guess I'm a little bit confused about how in vision we end up, as you were saying.
How in what?
In vision.
In vision.
Yeah, when the channel is open, sodium channel is always open, and then
Yeah.
cyclic D and P gets taken out. How does that start an action potential? Doesn't make any sense to me.
Okay, so let's take a look at that. It's a good question and it's a common one also. If we look in vision, how do we start an action potential? By closing a channel. All right, let's see, vision. Oh, I got immune system, don't I? Sensory system. Oh, blasted Ahern. Oh, it's here. Okay, that's why I'm confused. All right, so here's vision, and here's that signal. All right, so everything makes sense until you get to right here, right? All right, so right here it doesn't make any sense. Why? How does closing this channel do anything? Well, let's think about what's happening in this cell. Okay, your eyes I'll give you a little clue here. Your eyes burn a lot of energy. There's something like 25% of all the energy in your body is burned by your eyes. Did you know that? Okay, your eyes are using a lot of energy. Now, why are they using so much energy? You actually see it right there. What are they doing? Well, what are your other cells doing? They've got a sodium-potassium pump. What are they pumping out? Okay? Now, when we were pumping things out, what happens when we pump things out? Well, in the case of the mitochondria, we pump it out. We create a gradient. That gradient comes back in through complex 5 and makes ATP. All right? There's a link between the two. Here we're pumping out, and now it's coming back in. Right? So we're pumping out, it comes back in. The difference between the concentration of sodium out here and the difference in sodium in here is not significant. Okay? Because there's flow out, there's flow in. There's flow out, there's flow in. There's flow out. I close the gate, what happens? There's no flow in. I'm still going out. That's the potential. It seems totally cockeyed. It seems totally backwards. It doesn't seem like a very efficient way to do things. And I agree. I think it's one of the reasons that your eyes burn so much energy is because they're just wasting it, waiting, and then they close to generate the potential. Yes, sir?
Does this allow for that quicker response back? You said they sent 1,000% of signals.
I'm guessing it must, but to be honest with you, as I look at it, I have a hard time seeing that myself. Does this allow for that quicker response was his question. And I look at this and I don't know. I mean, vision is magical. Vision is hard to understand in that respect. But that's what it's doing. I mean, how is it that, not only do we have to close this, do we have to generate a potential, we also have to get this guy out of here. Okay? So a lot of things have to happen. They have to happen here very rapidly. But the answer is that they do. And that's surprising. Yeah, Jared?
I've heard that, and I can't speak for this mechanism, but I've heard that vision is pretty well conserved mechanism across a lot of biology.
Yeah.
And do you know if there's other variations of other animals, or maybe where they're from, that this would be advantageous? Some history based on it?
I don't know I'm not an evolutionary biologist. I don't know the answer to that question. His question was, are there differences in this mechanism across evolutionary biology? I think it's sort of your question. And I don't have the answer to that, no. Yes?
So I fully understand this mechanism, but could you sort of say that this is analogous in a way to the uncoupling protein?
Yeah, so is this analogous to the uncoupling protein? It's not unlike the situation we have with the uncoupling protein, yes. Not at all unlike what's happening with that. That's correct. Jared?
You just want us to know how the potential is created. We don't have to know the entire figure, or would you want us to memorize?
Well, I think, as always, anything I've talked about in class is fair game. But I think you should certainly know how the potential is generated. I mean, it's odd. It's really odd. Closing that gate creates the potential, and bang. And, when we think about nerve cells, they're exquisitely sensitive to potential changes. That's why they're so damn good at what they do. So closing that gate for an instant is enough to generate a voltage across this membrane, and that voltage across this membrane is all that's needed to start a nerve signal that makes it to the brain. So, and I guess it's also important to note that this is the initiator, okay? So this isn't how it's being propagated all the way along that nerve cell. This is how the signal is getting started. So we're not talking about a gigantic number of cells that are doing this. So maybe that's one of the ways in which it works efficiently. Giovanna?
So you said in taste there were the three that had empty membranes and then the other two. So the three that do, that's the gust-inducing or whatever.
So yeah, so the three sensory cells that are involved in taste that have a G protein are interacting with the 7-TM, yes. And let's see, that included, I have to think about the sweet, that included bitter, and that included umami. Is that right? Yeah, because the salt and the sour were the other. Yeah, right. Was that your question, or am I? Oh. Yeah, taste is sort of an odd one, isn't it? I mean, you've got different taste receptors. They function in different ways. And then we've got this capsaicin receptor that sort of lays over the top of all that, which isn't really so much involved in taste as much as it is involved in a sort of a pain sensation, which is kind of odd.
Can you go over recombination?
Can I go over recombination? Okay. Well, let's see what I've got. So when I talked about recombination, I was talking up here and didn't say a lot about recombination, but I will go through what I talked about before. Recombination, as I said in class, can occur by a variety of means. The easiest to understand and the ones that we most commonly think about are recombination between related sequences. And this particularly becomes, I think, more intuitive when we look at a figure like this. Actually, it's the next figure, where we see what's called strand invasion. Okay, so if we look at strand invasion, all right, here's these two DNAs. These guys are closely related. All right? This might be, for example, the red and the green receptors that we talked about with respect to vision, the opsins with respect to vision. Okay? They're closely related sequences. Now, closely related sequences, when you start invading a strand like you do here, okay, this strand is invading. We see actually it's sort of showing it backwards as we see here. It makes more sense if these base pairs can reasonably form that it's going to make a stabler duplex. If these are completely unrelated, it's really hard to imagine how in the world that's going to get in between something that's already nicely base paired. All right? So mechanisms that involve homologous recombination, the homology is the homologous genes. That is, they're related, the strand can invade, and it makes a pretty good job base pairing just like the original did. Maybe not identical, but very close to it right here. Okay? So the strand invasion proceeds, and one of the ways that the strand invasion, or not one of the ways, the way that the strand invasion resolves itself is through what's called a holiday structure. And this is taking that very same figure where we've had some strand invasion, and that strand has invaded across the system as we see here. It forms what's called a cruciform structure as we see there. And what actually can happen is there's unraveling and twisting. This guy unravels, and it's sending one of its strands over here. The other strand is going back up over here. And all these helices are twisting and untwisting as appropriate to accommodate the strand coming in or the strand going out, as we see there. Ultimately, at some point, something stops it, and there is an endonuclease that will actually cut. It will either cut it across here, or it will cut it across here. And that's a relatively random thing, how that will do that. Okay? Resulting in now two sequences that probably have some mispaired bases in there. They weren't completely identical to begin with, so they've got some mispaired bases in there. Well, what's going to happen when the cell sees those mispaired bases? It's going to say, bring in the DNA repair system. The DNA repair system is going to make its best guess at repairing those, and it may, since the DNA has already been synthesized, it can't tell which one's the parent strand. So depending upon which way it resolves that sequence, we're going to get generally new sequences. Okay? That's homologous recombination. Homologous recombination, like I said, you can see that strand invasion happening like that. There are non-homologous recombinational events that occur. One of those I talked about in class is those done by HIV. HIV integrates into the DNA. It integrates relatively randomly, which is one of the problems with it. You can't tell exactly where it's going to go. It goes many different places in the genome, and there's no sequences that it invades as such. And this enzyme that the HIV virus has called integrase facilitates that random integration into the genome. Okay? Liz?
Is integrase also used for homologous?
Yeah, it's a good question. So I kind of alluded to that when I talked about it in class. And I called the cellular enzyme integrase. And technically, the cellular enzyme is not integrase. It's actually something called a recombinase. But the integrase that does that integration is closely related to the recombinase that is involved in cellular recombination. Yes. Was that a question? Yeah.
I had a question about the eye cells you talked about earlier.
The eye cell?
Yeah, is the reason that our eyes close when we're tired because the eye burns so much energy?
Is the reason we close our eyes and we're tired because the eye burns so much energy? Yeah, I guess what I would say is it probably makes sense to not be firing those eyes. But I suspect that the answer is more due to the fact that if the eyes are open, the brain is processing information. And I think the brain is sleeping as much as the eyes are sleeping. So, but yeah, closing the eyes probably does save some energy, yeah. I don't know about you, but my brain needs to sleep every now and then. But you know, when you look what your brain does at night, you know, and you look at all that dreaming that you do and everything, the brain really is just doing something constantly. It's kind of a weird thing. Oh, sorry, Liz.
The
The Ames test, yeah.
Substance is highly mutagenic.
Yep.
Would you have it mutate twice such that you wouldn't get like a blue color?
Okay, so her question has to do with the Ames test, And the Ames test is dependent upon a mutation. That mutation restores a functional gene that wasn't functional before and gives some visible phenotype like a blue color or something. And her question is, well, is it possible it could mutate again so that you wouldn't get a blue color? Okay. Well, yeah, it's possible. Okay. But if you keep in mind that that were to happen, you would have to have a mutation rate so high that it would be going constantly, that's not going to A, result in viable cells if it's constant, and B, that not every time it mutates is it going to mutate a second time. That's going to be a relatively rare, even if you had 1 in 1,000, for example, which would be a very high mutation frequency. If you had 1 time in 1,000 that the replication was being mutated, you would see that. Well, that would mean that you would only detect 1/1,000 of 1/1,000th of those. So it's insignificant. It wouldn't show up. Yeah?
What the Ames test is, so you're just reactivating that one gene.
Yeah, that's right. So his question was, you're reactivating one gene.
Well,
Yeah.
no, but that's going to be a fairly random thing to reactivate that one base
Yeah.
pair.
Yeah.
So, like, that has to happen a lot for you to actually see the pattern.
Okay, so his question is,
What's the chance of your actual gene that's being reactivated being mutated beyond use?
I'm sorry, so say the last part. I'm not sure I understand the last part.
So you're reactivating this gene.
Yeah.
That gene, isn't there more of a chance of that gene being mutated beyond use than actually reactivating it?
Okay, so his question, your question is related to hers. So the question is that, well, I've reactivated this gene, but now maybe I can mutate somewhere else in this gene and inactivate it. Okay? Yeah, I can. But again, we're talking about very rare things that are happening. A very high mutation rate would be 1 in 1,000.
But
Okay?
how are we knowing that that's that one gene being, that one base?
Okay, wait. So remember what I'm doing in the experiment. What you're wanting to do is you're wanting to say, I'm trying to measure every mutation. I'm not. I'm trying to compare the mutation rate of this versus this. It's a relative thing. Okay? So I want to see what's it like if there's no substance, and I want to compare to what it's like if there is substance. Is there a difference between the two? Yes. Am I measuring exactly the mutation rate? No, I'm not trying to. I just want to compare nothing versus something. You see? And what you want is a quantitative measure. Okay? But the Ames test is aimed at giving you a simple answer in terms of, is this mutagenic or not mutagenic? Does that make sense? Okay. It's a common question again. So, yeah.
So, quick question back to recombination. This is like just recombination even in somatic cells, right? So it's not like the meiosis kind of recombination that you see.
So recombination can happen in any cell. Yes. Yes, sir.
Can you go over histones again?
Histones? Yeah, sure. So what did you want to know specifically?
Just kind of what their function is.
Okay, so if we go to histones, talked about that here relative to gene expression. And histones are these small basic proteins, basic in the sense that they have a positive charge at physiological pH. They have a lot of lysines and arginines in them. And because they're positively charged and because they have the shapes that they do, they can form a core around which DNA wraps. So a nucleosome core contains 8 proteins, 2 copies each of 4 different proteins, and the 4 different proteins are shown right here. They're called H2A, H2B, H3, and H4. And the structural similarity between these, I think, is rather striking, and they mesh together very, very nicely. Now, what happens is the wrapping of the DNA around these makes the DNA be more compact in size, which allows it to fit into the nucleus. It makes it harder for transcription and for transcription to occur. Of course, translation is occurring out in the cytoplasm. And that's why we had to have all those remodeling engines and things opening up those regions so that the transcriptional complex could get in there. DNA plus histones equals chromatin. Okay? DNA plus histones equals chromatin. If we take DNA plus let me just show you here. If we take DNA plus a core of 8, so this is a core of 8. There's 8 proteins right there in that bead. DNA is wrapped around it a couple times. If I go from one of these to one of these, that right there is called a nucleosome. So a nucleosome is a building block of chromatin. Chromatin is the result of all those things together.
So it's used as kind of a pack,
Packs it up.
Is it also kind of a protective thing?
It is protective. It does help to protect the genome, yes. Okay. Just this little segment right here, just the bead itself, is called a nucleosomal core particle. I didn't give you those numbers, those terms in class, so don't worry about that, okay? But that's what a nucleosomal core particle is. It's just the bead itself.
Is there kind of a relationship between the DNA that wraps around it, like specific DNA wrapped around the histone, or is it more of a-
That's a very good question. Is there a sequence specificity to the DNA wrapping around the histones? And people have studied this a lot. And it appears that the sequence specificity is in some cases, strong, and in other cases, it's not strong. Okay, so if you think about it, what you want to have is, let's say that you would like to have some spacing that would say, okay, we're going to have a regular wrap, and now we've got a new gene. Maybe if we could just kind of wait a little bit before we start wrapping, maybe we might allow access to some of these proteins that are in there. And so this is called spacing. And spacing is a consideration for the transcription of genes. And people have studied this, the question that you've asked, a lot because they would like to know, are there regions that favor those spaces that happen? It's a very, very interesting topic. Yes, Giovanna. Sure. Okay. So leucine zippers and zinc fingers, which is what she's asking about, are structures that we see in proteins that bind to DNA. We see these are listed here in eukaryotic cells. And the zinc, this is actually this is a leucine zipper. The leucine zipper is, the zipper part of it is the fact that if we were to go along this helix on the left, the one in yellow and red, okay, every time we went through a full turn, that is going from here all the way over to here, we would see sticking out of there a leucine. So every turn, right there, right there, right there, right there, it's actually in the middle of it, but let's say right there, right there, right there, right there, right there, right there, right there. Each time we see a leucine. Well, to go around one time is about 7 amino acids. One loop is about 7 amino acids. So every 7 amino acids, we see a leucine sticking out.
So you're saying that those are the histones, and then they're on the histones?
This is not histones. No, no, no. These has nothing to do with histones. Okay. These are transcription factors. Okay. Transcription factors are proteins that bind specific sequences in DNA. Okay. So they're not histones. No. Okay. All right. So here's all these leucines. Well, this guy over here has leucines on this side. And so the leucine side chains are relatively hydrophobic. They interact with each other very nicely and give stability to a structure like this. Up here, it's nice to hold it together. And down here, the ends of these structures interact with the DNA in a sequence-specific manner. Okay. So all that the leucine zipper is just a structure. Okay. It's just a structure that stabilizes a structure in DNA. Another structure relevant in, and these are all in transcription factors, another structure relevant in transcription factors are called zinc fingers. And zinc fingers, again, not in the histone, but we find them in transcription factors, has a structure where we have an arrangement of cysteines and histidines around a zinc ion that you can see right there. And that arrangement makes literally a portion of the protein that sticks out like a finger, and that finger actually inserts itself into the DNA. It's the part that reads the sequence. Yes?
Isn't there another one called a homeodomain?
There are others called homeodomains. You're correct. Yeah. There's a lot of different structures in transcription factors that bind to DNA. Yes, sir?
Are zinc fingers or leucine zippers found in DNA polymerases?
Are zinc fingers or leucine zippers found in DNA polymerases? No. Not to my knowledge, no. And again, these are really good for going there and binding to DNA. And a DNA polymerase, I want it scooting. I want it moving, right? So it probably wouldn't be a very good structure for a DNA polymerase, or an RNA polymerase for that matter. Yes?
So the zinc fingers and leucine zipper stabilize the DNA?
So, no. So the leucine zipper and the zinc finger are to stabilize a structure in a protein. And that structure in the protein binds to the DNA. Okay, so it's stabilizing a protein structure, just like we saw we had to stabilize structures, tertiary structures with ionic bonds, and we had non-covalent bonds, and we had all these various things. These are just stabilizing specific structures within a protein. And this specific structure is binding to DNA. Okay? So it's not stabilizing DNA. No, absolutely not. Yes, Liz?
One of the reasons you gave in class for the RNA polymerase being slower
Yes.
than the DNA polymerase is because it doesn't have a topoisomerase. But then if DNA gyrase is unwinding,
Yeah.
I guess I don't
Okay,
understand why
so,
it would slow it down.
all right, so I think you're confused here. So Liz said that one of the reasons I said was because that the RNA polymerase was slower than the DNA polymerase because it didn't have a topoisomerase. It's got a different topoisomerase setup. I didn't say it didn't have a topoisomerase, but I think what I said was it didn't have a helicase. So it's not peeling things apart at 6,000 RPM. Okay, that's a big difference. Even though there haven't been very many of you guys that have asked a lot of questions. Yes, sir.
Going back to histones, there was something you mentioned about unpacking the DNA by changing the charge of the lysine residue.
Uh-huh.
Can you go back over
Sure, sure. So his question has to do with basically putting acetyl groups onto lysines because that affects the packing of the histone with the DNA. This actually is what happens in the question that you're asking. So let me back up a little bit before I talk about this. So when we think about that relationship between the histones and the DNA, unaltered, that is just a native histone, a histone is positively charged, the DNA is negatively charged, that they're set up to wrap very nicely, and that makes a nice tight packing arrangement, and those structures just pack. That's how we end up seeing chromosomes in a microscope, is the fact that things are just very, very tightly and densely packed. To do transcription, or even to do DNA replication, we have to be able to get the polymerases access to the DNA so that it can go and either make RNA or in some cases, DNA. Well, for our purposes, we focused here on RNA. In order to do that, we have to basically loosen the bonds between the histones and the DNA. Okay? So the way, or one of the ways in which cells do that, is by putting acetyl groups onto the lysines of histones. Remember I said histones are full of lysines and arginines. Here's that positive charge of lysine. Here's an acetyl group. I put that on there. What was a positively charged side chain is now a zero side chain. Okay? So now this histone is going to be less attracted to the DNA than it was before, and it does what we refer to as loosening. Instead of being tightly packed, it's going to be loosely packed. And that loose packing turns out to be very important as we want to get access of transcription factors to that promoter sequence. Okay? The histone that has the acetyl group on the lysine is a target for binding by another protein structure called a bromodomain. And a bromodomain will recognize and bind to, okay, it will recognize and bind to an acetylated lysine. Well, let's imagine that I've got my transcription factor that has bound to a DNA. It's by whatever means, it's gotten to a sequence and it is bound to it. The example I gave in class was the estrogen receptor. Okay, it doesn't have to be the estrogen receptor, but it could be any. So it's bound to its target sequence. A coactivator protein binds to the transcription factor and binds. The coactivator is actually something that puts acetyl groups onto the adjacent lysines. So all the lysines around here are going to get acetyl groups put onto them, which means this whole area, as a result of this coactivator now being here, is going to get loosened up. Okay? In addition, we've now got acetyl lysines that are targets for proteins that have bromodomains on them. So those proteins that have bromodomains are now going to come and bind to those acetylated lysine residues, and one of them is this protein called a remodeling engine. Well, the remodeling engine takes this already loose structure. Remember, it's the histones have already been loosened their bond on the DNA, and it literally peels apart a region now so that all the other factors can come in and transcription can get started. Okay? So that's what's happening in that sort of mechanistic way in order to get transcription started in eukaryotic cells. Now, one of the things I didn't tell you, I described this in my molecular medicine class, and it's always of interest to people, it's really of interest to me, is when we look at proteins that are implicated in longevity, okay, how long you live, right? There's a group of proteins that are known that when you activate them in certain organisms, they make the organism live longer, okay? They're called sirtuins, S-I-R-T-U-I-N-S. And sirtuins, one of the things that they do, they take acetyl groups off of lysines. Well, what do? If you think about this, what they're doing is they're shutting off transcription. They're shutting down the cell. If you take and you activate a whole bunch of sirtuins, you're going to turn off transcription very widely across the cell. The less the cell is doing, the longer the organism lives. And some people have pointed to this as a means. There's actually a very cool phenomenon that's known for increasing longevity in organisms, where if you basically starve an organism for calories but still provide it with all of its necessary nutrients, you give it about 40% less calories than it wants to consume, it'll live 30% longer. Every single organism it's tested on, it's known. And what appears to happen when you're doing that is you're turning off an awful lot of transcription. And in fact, you're probably doing it by turning on the sirtuins. Yes, question?
I'm just curious, does something like that happen during hibernation or torpor?
Yeah, it's a good question. I don't know about that relative to hibernation, but it does occur relative to starvation. Not just calorie restriction, but also in starvation. It wouldn't surprise me if you didn't see something like that happening at least during the process of hibernation. But of course, hibernation has an end to it, and then things all get really going after that. So, yeah, I don't know.
So this figure you lost me with the remodeling agent. How does that interact with the acetylated?
Okay, so our question is, didn't understand the remodeling engine. So you understand we've acetylated lysine right here. So the remodeling engine has a bromodomain. So it binds to acetylated lysines, and now its job is basically clearing out the histones, opening up a bare area of DNA so that the RNA polymerase and other factors can come in and start transcription. So the remodeling engine is just kind of like clearing out a landing strip for a plane to come in. Yeah?
On the other side of this, say with reference to specific tissue types,
Yeah.
is there ever an instance where the DNA is tightened around the histones for, I guess, around certain genes that don't need to be expressed in certain cells?
Yeah, it's a good question, actually. So do cells have sort of chromosome-specific regions that are tightened that are tissue-specific? And yes, they do. They definitely do. And you can actually see these differences. Okay, you compare heterochromatin versus euchromatin. You see these differences in the microscope. Yeah. You know, I put that. I wasn't going to put that on the web page. Question if I'm going to be teaching this class again. Yes, I will. But I've actually recently taken on some new duties at OSU. I'm the director of undergraduate research now, and so one of the things I'm reducing in the process of assuming those duties half time are that I will not be teaching as many classroom classes. So I won't be teaching 450 in the fall. I won't be teaching 450 in the winter, which is going to be really weird. But then what I'll do is, like, every third year I'll be teaching one or the other. But this coming year I won't be teaching either one of them. That's just going to be weird. So I've been teaching them both for a long time. I love the class. I really love the interactions. And as I said in class, you guys have been, I'm not just saying it, the numbers say it, you're the best class I've ever had. I've never had, no, seriously, I've never had classes that had averages. You guys had an average, I think last term, one of the exams was like 77, and you've had 2 of them this term of 76, and that's just unusual. So either I'm getting soft in my old age, or you're a good class, and I think you're a very good class. So I think that's kind of cool. So like I said, it was a good class to go out on. Yeah?
In regards to longevity,
Yeah.
could that also be like a form to treat cancer? Would that like slow cancer?
So would using sirtuin slow cancer? That's a much more complicated question. I won't try that one. I guess, could it be a strategy? I suppose it could be a strategy. But one of the things that and I'm giving you just the sort of outlines of the action of these things. This is a sledgehammer. If we think we're going to turn off transcription very broadly, then we could imagine, as always, whenever we do with anything broadly in a cell, we can imagine there's going to be implications arising from that. This mechanism that appears to give survival to organisms living longer has evolutionary consequences. And the evolutionary consequences is we evolved this because we didn't always have food. We had to be able to survive a period of starvation, which is why starvation ties into this. We had to be able to survive a period of starvation, so when that happens, everything shuts down. Reproductive, in fact, the first place you see reproductive organs shut down. Okay? And that happens to conserve energy to get you through that whole process. Well, it's designed to get you through that process. It's not there to make you live longer. The byproduct is that if you do it long enough, you do live longer. But there probably are other consequences that happen with that. So the fact that you might use it as a strategy for treating cancer, yeah, maybe. But I would also, like I said, I also worry about sledgehammers when I'm doing things. So not good ways to kill flies. Yeah?
So has it been shown then, with people going through I guess you could compare to the Holocaust, that kind of starvation?
Yeah.
Do those people live longer if they made it through?
Yeah, the question is, if you have had starvation in your life, do you live longer? No, the answer is you don't. Okay, and so this phenomenon that I'm talking about, for animals where we make them live longer, it's called calorie restriction. It's different from starvation. So the consequences of starvation are that you starve to death or you make it through. To see it go over a lifetime, you have to not only have that calorie restriction like you see in starvation, but you also have to have the vitamins and nutrients that's necessary for normal living. Okay, so you don't see people who've had a period in their lives where they've starved that they live longer. No, that doesn't happen. But this phenomenon occurs every single organism it's been examined in. It works. There's actually people doing this right now, but they haven't been around long enough to tell if they're going to live longer or not. Cool stories. There's a lot of cool things happening with longevity. Like I said, in my molecular medicine class, we spend a couple weeks, maybe a week or so, talking about that. And it's a really fun discussion thinking about, at the molecular basis, what's happening and what we're rapidly learning. I mean, most of what we've learned about longevity has occurred in the past 10, 15 years. And we're still unraveling that. I shouldn't say we, I'm not doing it. I mean, we being biochemists, I guess I could be part of we. All right, are we done? The last, yes, sir.
Thank you for having me for your session.
Certainly, my pleasure. It's always nice to interact, and thank you guys for being a great class. You really have been a wonderful class. So, all right, shall see you at the exam on Wednesday at noon.
In our normal classroom.
In the normal classroom.